Hello, everybody. This is Gary Schwartz. I'm the Chief of Hematology and Oncology at Columbia University School of Medicine and Deputy Director of the Herbert Irving Comprehensive Cancer Center at Columbia. I'm delighted to have this opportunity to share with you today the latest advances in GIST as part of GIST Awareness Day. And I'm speaking to you from Columbia University, and this is being done in partnership with LifeDraft, who you are all familiar with, and has been a huge supporter of advances in GIST research and therapy for many years. So with that background, I'll start with the first slide. Precision medicine is a topic of discussion among all patients with cancer. The idea of precision medicine has evolved with the ability now to test the DNA within the cancer and to sequence it to determine genes that, in fact, are activated and make the cancer cell grow. This ability has transformed the field of medical oncology. Over 10 years ago, the screening and the sequencing of one person's DNA took about 12 months and cost well over $1 million. Now, with the advance of technology and high technology computers, as shown here, we can read the DNA much as we read a novel or a book to determine what the genes are present and which ones are abnormal within the cancer cell. This process takes about 7 seconds and now costs only several $1,000s, with the cost coming down as this technology continues to be developed. Now, in the area of GIST, we've known a long time that this ability to sequence the DNA, as shown in this slide, has resulted in the identification of 2 activating oncogenes. These are the genes that drive cancer. One is called KIT, and one is called PDGFR alpha, and they are abnormal or mutated in 93% of patients with GIST. Only 7% do not have this mutation. And for those tumors, they can have mutations in other oncogenes that have only recently been defined, such as SDHB, and then again BRAF, and other genes such as NF1. But the dominant oncogene is KIT and PDGFR alpha. These mutations are found in different exons. Exons can be thought of as the chapters within a big book. The most common exon for KIT is exon 11, and in PDGFR alpha, exon 12, but mutations can be found in other exons as well, and these are associated with different parts of these proteins that in fact are receptors that span the outer membrane of the cancerous cell. So in the next slide, we can look for mutations in KIT by sequencing. We also have an antibody, that is a protein, that can be used to stain the tumor cell. And as you see here, this is the tissue from a patient with GIST that was positive for c-KIT by this method called immunohistochemistry, or antibody staining, with the brown stain showing diffuse positivity. As stated, these mutations are quite present, and it was really with the advent of a drug that blocks these genes, especially KIT, but also PDGFR alpha, the drug imatinib, which is fairly specific for this particular gene, and this is a drug that has transformed the field of therapy for patients with GIST. I'm showing you here one of the earliest patients treated with imatinib on the first study, in which a randomization was conducted comparing 400 milligrams to 800 milligrams of the drug. As shown here, the patient had a CT scan. These dark gray areas that are covered by crossbars are metastases in the abdomen or the mesentery of this patient. And after a short amount of time, these lesions on the right CAT scan essentially resolved, and this patient has been on continuous imatinib now for almost 15 years with no evidence of recurrent disease, despite the presentation of metastases in the distant past. And this represents the unbelievable opportunity the targeted therapy has provided patients with GIST with use of imatinib or Gleevec to block this oncogene and have this type of dramatic effect. Now, the problem is basically what happens over time, and in this cartoon I'm showing you a GIST protein represented by the engine, which is what the KIT really is an engine for the cancer cell. And what happens initially with therapy is that Gleevec blocks this mutation in the gene and turns off the cancer cell and prevents the active engine from going forward, and the cancer cells stop growing. But over time, there are acquisition of other mutations, and with these other mutations, we try to develop drugs that will block them. So exon 11 being the most common mutation, these tumors may then develop mutations in other exons, such as exon 9, and therefore we try to develop drugs that block these other mutations, which represent bypasses over the most common mutation. So that reactivates the tumor, and we're back in action. The tumor starts to grow. With these secondary mutations, we have other drugs, such as Sutent or sunitinib, that again blocks it, or regorafenib again blocks it, and tries to prevent the engine from being activated. But the tumor still acquires additional mutations, despite the fact that we have initial benefit and turning off the oncogenes. But then a third mutation may develop, and again, we have new drugs, such as BLU-285, which blocks an exon 17 mutation, which is a rare mutation. However, in the setting of drug resistance, this mutation develops and again reactivates the cancer cell, as shown here, and allows the tumor cells to grow and divide. And the hope is that with these types of new drugs that block these secondary, tertiary, even quaternary mutations, that these new agents can block these mutations from turning on the GIST cell and provide opportunities for cancer patients to benefit from these new drugs targeting these other mutations that develop over time as part of a drug therapy. Well, this is one way to block a GIST tumor from growing. There's ongoing work, not just blocking the DNA, but if you look at the process, the DNA makes RNA, which makes protein, and there are drugs that are being examined to block the KIT protein, such as HSP9 inhibitors, and there's been some also interest in blocking the RNA that makes these proteins, and one drug that was tested in the past is flavopiridol, but other drugs may be in process that blocks not just the GIST oncogene, the KIT protein, but we have now the ability to block the RNA as well for future drug development. How important are these mutations? Are they significant to you as a patient? Well, they are. As shown here, this is a Kaplan-Meier curve looking at survival over time in patients with metastatic disease who were put on Gleevec or imatinib. And you can see that patients with an exon 11 mutation, that the survival here is extraordinary over many years, whereas patients with exon 9 mutations have a survival benefit that's less than exon 11 patients. And of course, in the absence of any mutation, the use of Gleevec has little activity and survival is not improved. So understanding one's mutational status is important. We encourage all patients to undergo sequencing of the tumor. This allows us to look at overall benefits of Gleevec, and also allows us to develop and personalize the therapy according to drugs that may better hit or block these specific mutations within the tumor cell. Now, there's been advances using personalized medicine in other cancers that have impact on patients with GIST. So I mentioned in the earlier slide that some patients with GIST have mutations that are not c-KIT, and one of them, in fact, is BRAF, R-A-F. This is an important mutation, is responsible for causing melanoma of the skin, and there's a drug that actually blocks this mutation that's present in 60% of patients with melanoma, and the drug is called vemurafenib. This drug has been tested in patients with metastatic melanoma. Shown here is a PET scan of a patient with wildly metastatic BRAF mutant metastatic melanoma. with the PET scan showing tremendous uptake in multiple bone metastases in this patient on the left. And after only 2 weeks of therapy, these multiple metastases have resolved. You can still see some uptake in the brain, which is normal on the top, and the bladder below, as well as the kidneys in between. But the majority of these bone metastases have resolved, and what now works in BRAF mutant melanoma can now be applied in BRAF mutant GIST as well. So opportunity is now developed by discovery of new oncogenes. They may be less prevalent in GIST, but their development and identification of these mutations in other cancers provide opportunities for patients with GIST who have also non-KIT mutations that are druggable with new drug therapies. We've heard a lot about precision medicine. Let's move on to another area, that of immunotherapy. And this is now a breakthrough area of medical oncology. Every night you can turn on your TV set and hear about Keytruda, Abrativo, and the advances in lung cancer. So what is all this about? Well, it turns out that we have a T cell that's present in our body that's been turned off over the evolution of man because if the T cell had been active, it would in fact attack the normal organs of the body, not just the cancer. And on the T cell there are immune breaks. One of them is called CTLA-4 and the other one's called PD-1. And these immune breaks turn off the T cell and inactivate the engine as shown in this cartoon here. And it basically is an immune brake just like the brake of a car. However, with new drug therapies, we have been able to show that we can target CTLA-4 with a drug called ipilimumab, shown here as IPI, and also PD-1 with two drugs, nivolumab and pembrolizumab, also called Keytruda and Abrativo. And by targeting these particular proteins, these new drugs release the brake, take the brake off the T cell, turn on the T cell, and make the T cell active against melanoma. However, the issue is, can these drugs work in sarcoma? We've seen great successes in patients with lung cancer, head and neck cancer, stomach cancer, even lymphomas. So the question has come, what about melanoma? What about sarcoma? But first, here's an example of a patient, in fact, with a type of lung cancer called KRAS mutant lung cancer. This is a tumor type that has been very resistant to any type of targeted drug therapy, despite personalized medicine. And again, here we have a patient with metastatic lung cancer, not melanoma, with diffuse metastatic disease. That area in the right lower part of the picture is nodal metastases. There's areas under the armpit that's elevated, diffuse disease. There's uptake in the brain that's normal. And after a short time in PD-1 therapy, you can see how this PET scan is cleared, and this patient has gone on to be cancer-free for a long time. One more thing about melanoma, we've been talking about that initially. This is the use of these drugs over the last 10 years. And you can see that at one time chemotherapy was the only drug that really had any benefit and had a minimal benefit in the treatment of metastatic melanoma. This is the survival data now that extends actually well beyond the 2 to 3 years shown here. But looking at this early data set, ipilimumab improved survival from up to almost 20%. With the PD-1 inhibitors, pembrolizumab and nivolumab, by 2013, survival now was about 40%. And the combination therapies of these two drugs, we're seeing survivals out at now 5+ years of between 50 and 60%, which is an extraordinary advancement in melanoma. Well, here's the first data now in sarcoma. It's a result of a study presented in 2016 and 2017 by Dr. Taube at ASCO. It's a SARC study, SARC study 028, in which patients with metastatic melanoma are given pembrolizumab, also called Keytruda, in the diverse number of sarcoma subtypes arising from soft tissue and bone. This is called a waterfall plot. Each patient is a bar on this graph, and you can see for patients who progress, the bar goes up. That's an increase in tumor size, and for patients who benefit, the bar goes down. And what's striking about this, of course, is that the majority of patients do not benefit from PD-1 or pembrolizumab targeted therapy. Only a small number do, and these tumors appear to be more common among a tumor type called a undifferentiated pleomorphic sarcoma, previously called malignant fibrous histiocytoma, now UPS rather than MFH. And these tumors appear to be highly inflammatory and therefore appear to be more sensitive to PD-1 targeted therapy. There are also a few patients on this graph with poorly differentiated liposarcomas, but the striking thing is the lack of activity for more common sarcomas such as leiomyosarcoma and the lack of activity for synovial cell sarcoma. Now, some of this data has recently been replicated in an alliance trial presented at ASCO 2017, again confirming the benefit of this particular approach for the UPS type of sarcomas and the lack of benefit for leiomyosarcoma. So why is this? Why are sarcomas not responsive to immunotherapy? Well, one of the reasons is the lack of mutations. This graph and this slide shows the response to PD-1 therapy according to mutational status. Now, melanoma is the very top of this, and melanomas have the most mutations seen among all cancers, and this is due to the sun. The sun itself is the most potent carcinogen known to man. Well, second down is lung cancer. Lung cancer also has a very high mutational rate in the tumor cell, and this is due to smoking. And as you go down this graph, you can see that the rate of mutations diminishes according to tumor type, and at the very bottom is sarcoma, which in fact has the lowest mutational burden, the fewest number of mutations seen in a tumor, and therefore this does impact upon the ability to respond to PD-1 therapy. Now, the other thing about response, and can we use this to advantage in sarcoma and ultimately in GIST, is a marker called PD-L1. I've been talking about PD-1 receptor being the receptor that's targeted by immunotherapy. Well, the protein that activates that receptor is called PD-L1. And the question that has come up is whether PD-L1 expression may in fact be predictive of benefit to therapy. On the far left here is a patient with lung cancer and a biopsy showing that we can use, again, this idea of immunohistochemistry or antibody protein detection to pick up PD-L1 on the tumor cell, and that's the brown stain as shown at the bottom of this histogram. Now, what was done in the study presented in the New England Journal of Medicine for lung cancer is they looked at the ability to be free of disease and not progress as a marker of expression of PD-L1 on the tumor itself. And going to the far bar, the green bar at 72 weeks, this study examined the expression of PD-L1 and found that patients who had over 50% of their cells being positive for PD-L1, in fact, were able to be on drug without progression of disease almost 40% of the time. But at 72 weeks, if you had less than 50% positive tumor cells, either even 0% or 1 to 49%, as shown by the blue bar or by the red bar, your chance of being on therapy at 72 weeks was almost zero. At 24 weeks, going further to the left, again, the therapy of PD-1 targeted drug therapy with pembrolizumab favored the patients who had over 50% PD-L1 positivity, and however, at 8 weeks it wasn't apparent. But overall, looking at this data, you would want to have a high PD-L1 expression to benefit from treatment. Without that expression, in fact, you would say there'd be little benefit to taking a pembrolizumab or PD-1 targeted therapy. So what about sarcoma? Well, we've looked at this in several series, and we've actually done this in cell line data. On the far left is the expression of PD-L1 by what we call Western blot, and it's broken down by tumor type, as shown here, where certain tumor types have very high expression, if there's a black line, and actually in some parts of this, there are tumor types such as leiomyosarcoma in lanes 12, 13, and 14, where there's really very little of PD-L1 expression, and as anticipated, patients with leiomyosarcomas have shown little benefit to targeted therapy with pembrolizumab or nivolumab. The far right of the blot shows how we can use these same cell lines and in fact treat them with a drug called interferon to actually raise the expression of PD-L1 from low to high, and that's just being shown in this plot here. For example, patient with leiomyosarcoma, this is a cell line, not the patient's tumor itself. This is from a patient grown in the laboratory. And you see these light gray bars, the expression is low, but when treated with interferon, shown here as IFN, the expression can become quite high. And this is true of many of the tumors we've studied, indicating there may be a way to treat sarcomas to raise PD-L1 expression from low to high with appropriate drug therapy. And these concepts of inducing or elevating PD-L1 are ongoing. What about GIST? And what about sarcomas in general? This is now not cell line data. This is actually immunohistochemistry data that is, again, using the antibody for PD-L1 and staining the tissue for a brown stain to see if we can pick up this expression in certain tumor types among sarcoma patients and not others. So B, in fact, is a patient with that tumor type called undifferentiated pleomorphic sarcoma, which responded to the PD-1 targeted therapies on the SARC trial and on the Alliance trial. And as you can see here, there's lots of brown stain reflecting high PD-L1 expression. In panel C, there is not as much expression in another patient with the same disease, again, UPS, indicating that even with the one sarcoma subtype, there's exceptional heterogeneity, where some patients with UPS may have high PD-L1 expression and others may have low. And finally, panel D is a patient with leiomyosarcoma, again, a subtype which there's been very little response to PD-1 therapy, and there is some staining, but not as much as in panel B. So it may be possible to use PD-L1 expression to predict benefit to PD-1 targeted treatments. Now getting to GIST, which is clearly the major thrust of today's talk. We've actually examined the GIST expression in this paper published by D'Angelo in Human Pathology to look at PD-L1 expression. And we've taken 29 patients. And you can see in the panel A, in fact, there's moderate expression of PD-L1 in 429 patients, as shown here. Panel B is the pleomorphics, again showing expression. However, even in GIST, in panel C, this is a patient with really no PD-L1 expression, absolutely cold by IHC. This is this protein assay analysis on tissue, and again with synovial cell sarcoma, really very little to no expression, again reflecting the lack of activity of pembrolizumab or nivolumab in this patient population. But yet, GIST, 29%, nearly one-third of patients having positive expression, giving us hope that in fact patients with GIST may respond to this type of treatment based on all the data published in lung cancer, melanoma, and other tumor types, showing us a potential prognostic biomarker. So this leads to this final slide of what do we do? Is it possible to treat these patients with GIST to benefit from this therapy? This is the randomized phase 2 study that was just presented at ASCO this year, showing the benefit of nivolumab plus ipilimumab, a combination of two drugs that is targeting both PD-1 and CTLA-4 in combination versus nivolumab alone, in the hope that maybe using the two drugs that release the immune checkpoints will be better than single-agent PD-1. The data from the study does suggest that two drugs are better than one drug. However, the two drugs with the addition of ipilimumab, the inhibitor of CTLA-4, does result in increased toxicities, which remains a concern, though in this sarcoma, we were able to give full dose of the nivolumab and only small doses of the ipilimumab, and this tended to reduce toxicity, but still did not completely ameliorate it, and this is a potential issue when we add the second drug. However, the goal is, based on the data of PD-1 expression, to actually open an arm for GIST. And over the next several weeks, there will be the first national trial ever to examine this issue of either single agent or combination data with two versus one drug in patients with GIST, based on the fact that there are patients who have PD-L1 expression, and we are hopeful that this will result in a new treatment options for patients with this disease. What about the patients with low PD-L1 expression? Well, going back one slide, again, this looks at the issue of PD-L1 expression for patients who have low expression. And what we found out in lung cancer and in melanoma, the patients with low PD-L1, in fact, when given the two drugs, their response data may be as good as those patients who have high PD-L1 expression. So going forward, there are other opportunities emerging in the field of immunotherapy. This is an ongoing issue of how do you look at biomarkers of resistance to Gleevec. Well, it turns out that one of the biomarkers of resistance is the induction of a protein called IDO. Now, IDO is involved in amino acid metabolism. It, in fact, converts tryptophan to another byproduct called calcineurin. This byproduct of calcineurin, in fact, has a negative effect on the T cells, those cancer-killing T cells of the body. And this process of tryptophan to calcineurin induction, which impairs T cell function, is actually activated in the setting of KIT resistance. So the concept has emerged, if this is a mechanism of resistance to KIT, and if calcineurin, in fact, decreases the effects of T cells, can you block IDO with a drug that would, in fact, result in the reversal of KIT resistance, would allow these T cells to become reactivated and therefore provide a new opportunity for patients with c-KIT or imatinib-resistant GIST. So this comes to now a new clinical trial that will be run nationally, combining a drug that blocks IDO, that is a drug that blocks the ability of the cell to move tryptophan to calcineurin that suppresses T cell function, and this will be done in combination with pembrolizumab in patients who are imatinib, Sutent, or regorafenib failures. And this is based on new emerging data about how we can regulate the tumor microenvironment and how this is specific for patients with KIT resistance. And we have great hope that this will provide a new treatment opportunity for patients with KIT-resistant GIST. Finally, you'll be hearing more today from Dr. Califano on this idea of targeting a systems biology approach to drug therapy. I don't want to steal his thunder, but the complexity of personalized medicine has resulted in a vast amount of data, not just in DNA, but in abnormal proteins and RNA. And the question is, how do we integrate this data? Can you look at it all across multiple platforms and find commonalities that are in fact druggable? And what Dr. Califano has shown and proven is that you can look at this data and find that many of these systems, in fact, narrow down to one or two bottlenecks, that the proteins, the RNAs, the DNAs all signal in such a way from high up, but however, there's a commonality that they all share, and he calls these master regulators. So rather than targeting the multitude of upstream events and the complexity at this level, we should then be able to target the bottlenecks from which all these pathways converge. And we're working closely now with the LifeRaft group to identify what these bottlenecks are. We're working closely with them to use patient tissue from the LifeRaft tissue bank. We're using this tissue to look at the multiple pathways that are activated and looking for the commonalities, the master regulators that are the bottlenecks of these tumors, and then find drugs that block them. And based on this data, we hope to generate a phase 2 trial for patients with imatinib and KIT refractory resistant GIST, and begin clinical studies based on master regulator targeting over the next several years. This could be transforming. It's using personalized medicine to its ultimate degree, but trying to organize it in a fashion that allows us to identify new drug therapies for patients with GIST-resistant tumors. So in conclusion, we're moving forward with great alacrity in the area of GIST with new clinical trials. All this development requires clinical trial participation. I encourage all patients with GIST to consider this and these studies as they open across the country, both with immunotherapy, precision medicine, and master regulator targeting. It's the precision medicine and immunotherapy that I believe offer patients with GIST new treatment opportunities. We've learned lessons from other cancers, such as the BRAF story, and lessons in immunotherapy from lung cancer and melanoma, and these can now be applied to GIST. It's the understanding of biology and immunology that have provide new treatment opportunities. And I think by partnering with groups such as LifeRaft, who have been patient advocates for many, many years and have provided opportunities for patients where none existed before, this academic partnering with LifeRaft will allow us to conquer this disease and provide a new future for patients with GIST and their families as well. So thank you very much.
So good morning. I'm Andrea Califano, a professor of chemical system biology at Columbia University. I'll be talking today about some new system biology approaches that we've developed, and in particular the application to imatinib-resistant GIST. Just a very quick disclosure, as I'm on a scientific advisory board of Cancer Genetics and was the founder, stockholder, and chair of the MSAB of Darwin Health, is a new company that is using some of these technologies, so I wanted to have everybody to be aware of the potential conflict of interest. So very briefly, just before we get into it, I have to kind of summarize in a 101 lecture in biology in one single slide, just to make one very important concept clear. Our cells in our bodies all have exactly the same DNA and yet end up doing very different things. And so one question that many people may have is how it's possible that cells that are so identical at the genetic level can do things as different as being a liver cell or a blood cell or a neuron. And the reason this happens is because our DNA is just a template, and that template makes another molecule called RNA, or ribonucleic acid, and then finally that molecule ends up making yet another molecule called a protein, and the proteins are actually what get things done in the cell. And so depending on the state of the cell can actually end up making very, very different RNA and very, very different proteins. And that's how you get the same DNA to produce states of the cell that are very different. This is very important because the problem is that when you have a mutation inside the DNA molecule, that mutation gets very faithfully transcribed into the RNA, and the RNA puts that mutation into a protein. And so the protein now starts misbehaving. It doesn't work as it was originally planned to do. And so, for instance, this is an example of a mutation in a very important oncogene called KRAS, which is a major target for sort of attempts to create new molecules for therapy, and that mutation disrupts the normal behavior of KRAS. So what's the big idea that led to initial approaches in personalized medicine, and in fact, very effective even in GIST sarcoma? The big idea was that if we build drugs that target the proteins that have these activating mutations, these mutations that make them behave in a way that is really not normal, then maybe we can shut them down and we can actually cause the tumor to lose its key element of the engine that makes it spin. And so this has worked quite well and has had quite a number of successes. But unfortunately, as we're all very aware of, there are also some limitations. And so today, I'm going to be talking more about what these limitations are and how can we address them using somewhat a complementary approach. The key and most fundamental limitation is that almost all targeted therapy works for a while and then is unfortunately followed by relapse with a form of the tumor that is now drug resistant. And this is a problem because once you have now exhausted your ability to fight against that protein that has the specific mutation, now you're left with very little that you can work in terms of fighting this new version of the tumor that presents. And even worse is the fact that many tumors, in fact, lack even that initial mutation that is actionable, as we say, that is, can be targeted with a drug, and therefore they don't even respond to the first class of targeted therapy that we could use. For instance, an example in GIST sarcoma is that many GISTs have mutations in two proteins, c-KIT and PDGFR alpha, and this is now sort of started as a spur of activities in using inhibitors that were originally designed for KIT-enabled kinases or PDGFR alpha to be used as therapeutics in GIST. And unfortunately, many GISTs don't have those mutations and therefore will not respond to these drugs. And even the GISTs that actually have those mutations, in some cases will actually either not respond or most likely fail to respond after a while and present a form of the tumor that is no longer treatable with those inhibitors. So the question is, how can we do something that complements and extends this type of paradigm by digging a little bit deeper in the genes? And so I want to present one image. So as I said, the cells make this molecule called RNA, and the RNA makes proteins. So if you actually look at the RNA of different tumors, in this case what I'm showing you here on the bottom graph is about 20,000 tumors that have been collected in a large database called the Cancer Genome Atlas. And what you see here is quite remarkable because it shows that some tumors, for instance, what I'm showing you here is a group of tumors called triple-negative breast cancers and another group of tumors called luminal breast cancers, are essentially identical when you look at their RNA. Right? They look exactly the same, and that's why they cluster so tightly when you actually look at their RNA structure. And in fact, if you look at here on this graph, if you compare even tumors, which are the blue graphs, to normal tissue, which is the red graph, you see that they're almost indistinguishable in terms of how tight the distribution of the various samples are. And yet, if you actually look at the genetics of these tumors, so for instance, these are the mutations that are present in triple-negative breast cancer, they're literally all over the place. There are virtually no two patients that have the same genetics. And this was a very important observation. It's essentially a paradox. How is it possible that tumors that are so different at the genetic level end up being so similar, so virtually identical in the way they actually make RNA, they make proteins, and in fact, in the way they maintain their state as a tumor. And this paradox was essentially the motivation that prompted the line of research that we have developed here at Columbia, which basically has tried to explain this paradox using, if you want, an actual mechanistic approach. And the idea is actually quite simple, and it complements what has been known as the oncogene addiction hypothesis, which is what has led to the development of drugs like imatinib for GIST sarcoma with KIT mutations. And the idea is that if you end up having the same state of the cell, even though you have completely different mutation, there has to be some place in the cell where the effect of this mutation is actually integrated, like a bottleneck. So everything is now collected by this bottleneck, and then that bottleneck controls the state of the cell. And so what we have done, we have demonstrated the existence of these bottlenecks in many, many different tumor types, from glioblastoma to leukemias to prostate cancer to follicular lymphoma to breast cancer. And we have actually shown that these bottlenecks work exactly like the logic that allows a cell to develop into either a liver cell or a brain cell or a blood cell. That is, they maintain the state of the cell in a largely independent of the DNA state, largely independent of the specific mutations that induce the tumor, and that's why such different patterns of mutation can produce the same tumor. So we have called the very small number of proteins that sit in this bottleneck and integrate all these mutational signals, we call them master regulators, and we called the little module that they form a tumor checkpoint. Why is a checkpoint? Because like on a highway checkpoint, all the bad signals that come from mutation have to go through it in order to implement the state of the tumor. And so we say something very simple, which is that right now, what we're actually doing, we're trying to drug the oncogenes. And the oncogenes are up here, very upstream of the bottlenecks, and somewhere in this sort of the upper side of the hourglass. And when you do that, all you're doing, you're selecting for cells that do not respond to those drugs because either they have other mutations that bypass the activity of the drug, or in fact depend on completely different mutations. And so what you're doing is really like in evolution, you're selecting for cells that no longer respond to the drugs, and these cells will grow independent of the drug. But if you now develop drugs to hit the tumor in the bottleneck, then it becomes very difficult for the tumor to actually bypass that because there is no alternative path. You have to go through that bottleneck. And so it's a little bit like saying that cancer is like a colander and there's water coming out of all these holes that represent mutations, but there's a natural funnel underneath the colander, which you're seeing here on the right, and if you put a cork in that funnel, you stop completely the flow of water without having to worry about plugging every single hole. All right, so this sounds great in practice, but how do we find these proteins? Well, proteins are like generals. Each protein controls a bunch of other genes, and they can either tell them, like in a war, to stay in reserve and not move, essentially be on the quiet side, or they can tell them to go and attack the other army. And so this is exactly what we're seeing here. This is a very important general in many tumors called EGFR, epithelial growth factor receptor, which is a close parent of the PDGFR alpha, which is activated in GIST and mutated. And so if the targets of these particular proteins are not particularly differentially expressed, that is, the positively regulated one, the activated one, are not very different from the ones that are inactivated, which are shown as black lines, then we know that this protein is not very active and is not contributing to implementing the state of the tumor. But if you look at the bottom graph, where all the ones that are controlled in a positive way are all activated, and all the ones that are repressed are all inactivated in the cell, which we can measure very efficiently using something called, by sequencing the RNA of the cell very, very cheaply and effectively. Now you know that EGFR is very active. And so we can basically ask, what are the proteins that are maintaining the tumor state by simply looking at their targets. And we've done this for many tissue types, including GIST, sarcoma, and we've done this for about 6,000 proteins that include all the proteins that are involved in cancer. So now we can very simply go from a really, truly inexpensive, talking about a couple $100 RNA sequence of the tumor cells to knowing exactly what are the proteins that sit in those bottlenecks. And let me give you an example. This is actually from a clinical trials which we're now running in breast cancer, which has just moved to phase 2 because of very exciting results in phase 1. And the idea of this is a tumor that is started from a mutation in the protein called ERB2 or HER2. And what happens is that there is a drug called trastuzumab that completely blocks the activity of that protein, and it works in many patients. About 70% of the patients with this mutation actually respond to the drug. But unfortunately, about 70% of those who respond will eventually relapse, and that drug will no longer work. And the reason it doesn't work anymore is because there's another set of proteins that sit in the bottleneck, that sit in a tumor checkpoint. One of them is called STAT3. And what this protein does, it activates another gene called IL-6. This creates a protein that goes outside of the cell, activates a receptor on the surface of the cell called IL-6 receptor, activates a protein called JAK, and that JAK activates that tumor checkpoint. And so there's a loop, as you can see here. So even if you block the signal here, you have this little loop that keeps going. It's a little bit like a diesel engine. So if you now combine two drugs, one hitting the ERB2 protein called trastuzumab and one hitting the JAK kinase called ruxolitinib, now you have dramatic response. And in fact, some of these patients have had really dramatic responses and significant life prolongation. So now the question is, how do we target this tumor checkpoint? These are a bunch of proteins. We don't know exactly how to do it. So remember that what I told you is that using our approach, we can now measure the activity of proteins. So we can do it either in the naked tumor, or we can do it when the tumor is being exposed to a drug. So we can ask, is this protein active? And by the way, what happens to that protein when I put this drug in the tumor? So what we do is we take a sample from a patient. We get the RNA measurements by sequencing the RNA in the cell, as I said, very cheap and very rapid. And then from that, we can figure out all the bad proteins that have been activated and all the good proteins that have been inactivated. And now we can simply test all the possible available drugs, including all FDA approved and all drugs in phase 2 and phase 3 clinical trials. It's about 400 drugs. We can put them in a tumor cell, and we can ask what happens to the RNA of that tumor cell, and then from that, what happens to the proteins that were apparently activated or inactivated in the patient. And so what we want to find is a drug that actually completely reverses the activity of these proteins. The ones that are up now are going to be down after the drug has been used, and the ones that were down are now going to be up. And the reason this works is because these proteins all control each other. And so they're like an on-off switch. When you hit the right entry point, that entire module of protein will reverse its activity and shut off. So we now tested this in a very exciting trial, which we call the N-of-1 study at Columbia, where we are enrolling about 260 patients. We're about halfway there. We've enrolled 95 patients, and these patients have been implanted, many of them, into a mouse model. So the mouse is actually carrying a piece of the patient tumor that was directly transplanted from the patient into the mouse. And this is basically the closest you can possibly get to having a model of the tumor that you can experiment with drugs on that is actually not the tumor itself in the patient, because it's very difficult to experiment with drugs in a human being. It would potentially have disastrous consequences. So this gives us a model that we can test all the things that are predicted by our methodology. And the way we do it is we basically take a biopsy from a patient, we transplant it in the mouse, we profile the RNA both in the patient and in the mouse, we do our analysis of what are the proteins that are either hyperactivated or hyperinactivated, that is the tumor checkpoint proteins, both in the patient and in the mouse. And then we predict which drug would work based on the approach that I just showed you. And we test them in the mouse model, and we also test what happened to the mouse model at a very short time, that is before the drug has actually killed the tumor. And the reason we do that, this is called pharmacodynamic studies, this box here, is to figure out whether the drug actually killed the tumor by hitting some strange mechanism that we didn't even think about, or it did so by actually reversing the activity of the tumor checkpoint. So this is what you could call the money slide. In fact, these are we're now down 7 different, what we consider untreatable tumors that have failed somewhere between 3 and 6 targeted therapies or chemotherapy, essentially lines of therapy. And what we did for these patients that, as you can see, are in very, very different tumor types, from a imatinib-resistant GIST sarcoma all the way to a metastatic KRAS mutant colorectal cancer. We transplanted these tumors in the mouse, we predicted the drugs using our approach, and we tested somewhere between 1 and 7 drugs actually in each one of these models. We gave the drugs to the mouse basically, and we looked at what happened to the tumor. So the way you read this graph is that on the vertical axis you see the size of the tumor. 0 doesn't mean that the tumor has no size. 0 means that it's a starting point. Basically, the tumor was about 1 cubic centimeter. And then the y-axis presents the volume, so you're going increasing or decreasing the volume size. The black line represents the untreated animal, an animal that was given just a control substance that was not affecting the tumor at all. And you can see basically these tumors are all growing very quickly in that control arm. And then the colored arm show over time what happened to the tumor size once you gave it the drug that were predicted by the algorithm. And what you see what is quite remarkable here is that these are all tumors that essentially were considered to be untreatable, should not have responded to any drug. In fact, you can see here in this pancreatic cancer, we treated the tumor with 2 drugs, not predicted by the algorithm, but actually decided by an oncologist, and neither one of those worked at all. While on average, more than half of the drugs that were predicted by the computer induced what is called an objective response. That is, either the disease remains completely stable for up to 30 days. 30 days in a mouse corresponds to a much, much longer period of time in a human, of course, or they actually were significantly reduced in size. So for instance, here you have 2 drugs, fludarabine and topotecan, that had quite a striking effect on this imatinib-resistant GIST sarcoma, and these are drugs that you would have never given to a GIST sarcoma. And the same thing you can see here with selinexor in a prostate cancer, irinotecan in a triple-negative breast cancer, selinexor in a rhabdoid tumor, etc. And several of these patients were actually treated, and 3 out of the 4 that we treated had quite striking responses. For instance, this patient here, after having failed to respond to any therapy, was treated with irinotecan and was stable for about 8 months. This patient here with anaplastic meningioma, after essentially having a tumor that was growing uncontrollably on any kind of medication, was absolutely stable for more than a year on a drug called etoposide. So this is promising. As I said, about 16 out of 30 of the drugs that we tested were in fact responded, the patient responded. And remember, these drugs were not actually screened in the mouse model or in cell lines. They were predicted by the computer to be the best answer to that specific tumor type. In fact, many of them are drugs that you would never typically use in the context of the tumors in which they were predicted. Equally important, we showed, this is for the GIST sarcoma, that when you actually took an early sample after 3 days that the mouse had been treated with that drug, in fact, the drug did reverse all the master regulators of the tumor. So the red bars are all the proteins that are the bad proteins that are hyperactivated in the tumors, and you can see they're all on the left side of this graph. They've been shut down. The blue bar were the good proteins that were down in the tumor, and now they're very highly activated. And essentially, this graph shows you how effective the drug was in creating that inversion. And so all of the 5 drugs that actually worked and had an effect on the tumor have these patterns where all the red bars are on the left, all the blue bars are on the right. The one drug that didn't work didn't do that, and you can see the red bars now are on the right and the blue bars are on the left. So it did not reverse the activity of these bad proteins. So just to summarize, the idea here is that cancer is like a very bad and very complex computer algorithm, and we need to use math to figure out how it works. In some cases, the solutions are simple. We can use a single protein that has a mutation that we can find in the DNA, and magically there is a drug that we can use to target that protein. But in most cases, things are a lot more complicated, and so we have to open the box of cancer and look inside. And this is an editorial that I wrote for the Wall Street Journal with a colleague of mine to actually make that point and start to foster more research in that direction. And just wanted to thank quite a number of people, including many of my collaborators on this research, in particular Gary Schwartz, whom you will also hear from in terms of just sarcoma, a sarcoma specialist, and also wanted to thank the Libraff Foundation because through a collaboration that we've done with Columbia University and New York Presbyterian, we're now really moving forward to applying this kind of novel concept, if you want, complementary concept to targeting mutated genes in the context of imatinib-resistant and otherwise drug-resistant sarcoma. So thank you, and